Welcome. Today, we're diving into advanced statistical approaches for determining expiry dates. I'm Dr. S.K. Singh, and together we'll explore how data and analysis can help us predict product lifespans more accurately. Let's get started and see how these methods can make a real difference. Let's start with why expiry date determination is so crucial in pharmaceuticals. It's not just a date on a package, it's a decision that impacts safety, effectiveness, and compliance. A well-justified expiry date protects patients, supports the supply chain, and meets regulatory demands. Today, we're seeing advanced statistical methods play a bigger role, helping us estimate shelf life more accurately. The ultimate goal: to make sure all critical quality attributes stay within approved limits for the entire product lifecycle. When it comes to setting expiry dates, there are several key regulatory bodies we look to for guidance. These include the International Council for Harmonization, the World Health Organization, the FDA in the United States, the European Medicines Agency, and the Pharmaceutical Inspection Cooperation Scheme. Across all these organizations, the expectation is clear: expiry dates must be grounded in solid science. That means using valid stability data, applying the right statistical methods, understanding how each product degrades, and incorporating risk assessment principles. This ensures safety and reliability for everyone. Statistics play a crucial role in figuring out expiry dates. They help us evaluate how stable a product is over time, spot differences between batches, and understand how quickly it degrades. Beyond that, stats let us predict future product quality, anticipate when specs might fail, and set realistic shelf life limits. They also support important tasks like extrapolating shelf life, deciding when to retest, and preparing regulatory submissions. Ultimately, the goal is to turn raw stability data into expiry dates that stand up to scientific scrutiny. Let's talk about linear regression, a common tool to estimate how something degrades over time and to predict what might happen in the future. It's straightforward and often used under ICH-Q1E guidelines. The key assumption here is that the quality changes in a straight line, steadily over time. But that's also its limitation. Real-world degradation can be more complex, and linear regression might not capture those nuances. This table shows a typical example of assay results declining gradually over 2 years. Now, let's dive into regression-based shelf life prediction. This approach uses historical data to find patterns, linking product age with quality changes over time. By fitting a regression model, we can estimate how long a product will stay fresh or effective. It's a powerful way to make informed decisions about expiration dates, reducing waste while keeping safety front and center. In short, regression helps us predict shelf life with more confidence and clarity. When regulators look at shelf life, they want us to account for uncertainty, not just rely on average predictions. That's where the confidence interval approach comes in. Instead of just using the mean, we consider the lower and upper 95% confidence limits. The expiry date is set where this interval crosses the specification limit. This method gives us extra confidence, ensuring the product quality stays reliable throughout its shelf life. It's a smarter, safer way to define expiry. Now let's talk about poolability analysis. This is all about figuring out if we can combine stability data from different batches. We do this by comparing key factors: the slopes, intercepts, and variability between batches. For example, if we have 3 validation batches, A, B, and C, and their degradation rates are statistically similar, we can pool their data. Why does this matter? Because pooling leads to a more robust and reliable shelf life estimation, which regulators appreciate. Let's talk about analysis of covariance, or ANCOVA, a powerful statistical tool we use to dive deeper into data. ANCOVA helps us understand how different factors influence outcomes over time. For example, it evaluates time effects, batch effects, and their interactions. Imagine tablet batches made at different sites. ANCOVA tells us if these batches degrade differently as time passes. This insight is crucial, especially when deciding if separate shelf life assignments are needed. It also plays a key role in regulatory decisions, backing up scientifically sound pooling strategies. Let's talk about nonlinear degradation modeling. Many products don't degrade in a simple, straight line over time. Think about biologics, vaccines, protein therapeutics, or modified release formulations. Their breakdown can be quite complex. To capture this, we use models like exponential, Weibull, polynomial, and logistic. These aren't just fancy math. They help us predict how these products change with much better accuracy, especially when the degradation doesn't follow a predictable pattern. This approach gives us a clearer picture of product stability. Bayesian statistical approaches are gaining attention from regulators because they offer a smarter way to handle data. They combine what we already know, historical knowledge, with current stability data and expert judgment. This mix helps us make better predictions, even when data is limited. It also gives us a clearer picture of uncertainty and lets us adapt shelf life estimates as new information comes in. For example, if we have a platform formulation with lots of past stability data, we can use that to confidently predict shelf life for a new strength. When we look at traditional methods, they tend to focus on just one quality attribute at a time. But multivariate statistical modeling takes a different approach. It evaluates multiple factors all at once, things like assay, dissolution, impurities, moisture content, and physical properties. To do this, we use techniques such as principal component analysis, partial least squares, and multivariate regression. The real benefit here is that we get a holistic view of stability, a more complete picture that helps us understand how everything interacts together. Now let's look at how AI and machine learning are transforming stability prediction. We're predicting key outcomes like degradation pathways, shelf life, and failure probability. To do this, we use advanced models, random forests, gradient boosting, support vector machines, and neural networks. These models take in a variety of inputs, including stability data, process parameters, packaging details, and environmental conditions. The future points to predictive stability intelligence, smarter, faster, and more accurate insights that help us stay ahead. Let's look at how we used statistical evaluation to determine the expiry date for this film-coated tablet. We ran a linear regression on stability data, applying a 95% confidence interval to ensure accuracy. Poolability across 3 batches was confirmed, meaning the data sets could be combined reliably. The model predicted a specification failure at 43 months, but the regulatory decision set the approved expiry at 36 months, providing a solid safety margin. This approach balances science and safety in determining shelf life. To wrap up, we've explored a range of advanced statistical tools that are reshaping how expiry dates get determined. From linear regression and confidence intervals to ANCOVA and poolability assessments, and beyond, into nonlinear modeling, Bayesian statistics, multivariate analysis, and even AI and machine learning. These methods turn expiry date estimation from a simple trend check into a rigorous, data-driven science. The result: more accurate, reliable shelf life decisions that boost product quality and keep patients safer. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.